So good morning, everyone from Canada. My name is Hui, and I'm glad to coordinate this series on risk assessment, and we are dealing with questions regarding environmental and human health issues. So today we got the last lecture in the series, and I'm glad to have Professor Lynch from the prestigious University of Birmingham to give us a lecture on how safe are nanomaterials. So let me just give you a bit of idea about the background of Professor Lynch. So she's actually a professor of environmental nanosciences from the University of Birmingham, and she undertook her PhD in physical chemistry from the University of College Dublin, and she got as well her MBA and bachelor in chemistry as well. And she has published more than 145 publications. She has written and co-written 10 book chapters and one book. Her work is very high impact, so she has received more than 12,000 citations on her work. So it's something very amazing. And on top of that, she's doing editorial work and activities. So she's currently an associate editor for Environmental Science Nano, and she's as well in the editorial board member for the online journal Particles and Fiber Toxicology. Her research interest, she's focusing her work on the synthesis of novel composites for biomaterials and environmental applications. She's working as well on the fate of these nanoparticles in the environment, notably on the interactions with biomolecules, on the fate and behaviors of nanomaterial in the environment, and evaluating the impact and the risk of nanomaterial in the environment. So I won't take longer, and I believe that we all expect and look forward to her lecture. So thank you very much again, Professor Lynch, for your acceptance giving this lecture to us, and then we are all looking forward to listen to you. Thank you very much.
Okay, thank you very much. And I can share my screen.
Yes, please.
The green button. Yes. Okay. Perfect. Okay. Did that work?
Yeah. Wonderful. Thank you very much.
Perfect. Okay. And now I just need to move that one so I can put it into screen mode. Okay. So, hello everyone, and thank you very much to have the opportunity to talk about my research and to tell you a little bit about nanomaterials and what we're doing to assess how safe they are. So I gather some of you are coming from all sorts of different time zones and different countries, so fantastic to have you all here and fantastic to be part of this, this lecture series. So with no further ado, I'm going to start to talk to you about what nanomaterials are, why we're interested in them, and some of the specific challenges they present from the viewpoint of exposure, hazard, and risk assessment. So nanotechnologies and nanomaterials are everywhere. As a consumer, you wouldn't necessarily go and buy a nanomaterial, but you would buy many products that have nanomaterials in them. So this figure here, which is quite an old figure from 2009, but it sort of summarizes the range of areas where there are commercial applications of nanomaterials and nanoparticles. So they're in textiles, they're in biomedical applications, in healthcare, in food and agriculture, industrial, so in terms of pigments, in terms of energy capture, all sorts of things. In electronics, so your mobile phones and your laptops all have nanotechnology and nanoparticles in there. But also a wide range of applications in the environment. So as environmental catalysts in wastewater treatment and in renewable energy. So as for energy storage, energy capture, harvesting, etc. So nanoparticles is such a general term, but it talks about materials that are in the size scale of nano. So depending on what definition you look at, that can be anywhere from 1 to 100 nanometers or even 1 to 1,000 nanometers. So it's many applications, very many chemistries. So a lot of the elements from the periodic table can be produced as nanomaterials. And I'll show now some examples in a few minutes. They exist in many forms, so many shapes, sizes, complex architectures, mixtures, and so on. And some of the other issues are that we need to think about whether they're free or embedded. So in many of the products that I've mentioned, they would be embedded in a polymer matrix or into some other material, and therefore we might not be directly exposed to them, compared to ones that are free and, for example, in an aerosol that we might breathe, or a sun cream that we might apply to our skin. So free versus embedded is an important concept in terms of thinking about nanoparticle toxicity and risk. So the unique features of nanomaterials, what makes them so exciting and so useful for that wide range of applications? The main point about them is that they have a disproportionate amount of their atoms are present at the surface. So if we take a big particle such as this one here, the atoms in red are the ones at the surface. Obviously this is a two-dimensional representation of 3D, but if we start to break that down into medium-sized particles, you can see already that the number of the red atom, or the amount of red, increases very, very rapidly. So here we have already 20 times more surface area for the exact same mass as we had here. And then if we go smaller again, if we make them tiny, again, the same overall size, but now we have 60 times more surface. So much of the material is now surface. And this brings all sorts of new and interesting properties, and particularly where the surface dominates over the bulk. And these surface atoms are all highly reactive. So that's what makes them so useful for catalysis, or for as sensors for binding things, for energy capture, and so on. That these surface molecules or surface atoms are very active. So just a quick snapshot, since I work mostly in environmental area, I just thought I'd show some particular examples of the use of nanomaterials in the environment. So they're being developed and used widely for purification of water, at low cost purification of water, but also for desalination. They're used a lot for pollution reduction and remediation, again because of that high surface area that makes them very sticky to other pollutants that can bind to them. So they've been developed for remediation of heavy metal pollution, or for remediation of hydrocarbons, or all sorts of other pollutants. Used very widely in energy capture and storage. So solar panels, for example, use titanium dioxide nanomaterials, and also applications for carbon capture and nitrogen capture, and influencing the biogeochemical cycles. There are also a whole range of new materials that bring with them new properties. So, for example, self-cleaning windows, which are increasingly being used in skyscrapers and tall buildings, again, use the superhydrophobic properties of nanoscale materials, which basically means that the droplets either don't stick at all and run off, or they spread so well that you don't get droplets at all. You just get a continuous film. They're also used very widely in cars, and in actual fact I got an email in my inbox this morning inviting me to a conference on the use of nanotechnologies and nanomaterials in Formula One. I was like, well, that's kind of interesting. It's a bit away from what I do, so I reluctantly have to say no, but I thought it was a pretty exciting area. I'm sure I could tempt some of my students to go along to that. So they're used very widely in automotive and aerospace industries to make materials that are cheaper and stronger, and very especially lighter, so greater fuel efficiency. An interesting one is they're also being used to make sort of active or responsive materials. So, for example, a nano cement, where it will exist in one phase or structure until there's some structural damage or some shearing, and then it would self-repair by changing structure or by interacting. And then they're also used quite a lot because of the really, really bright colors of nanomaterials. They're used quite widely for art conservation and restoration. So for those of you that don't work with nanomaterials, one of the other interesting properties they have is they have quantum confinement of electrons. So that means then that they have quite different and bright optical properties. So for example, gold, which in the bulk we know is sort of the yellowy gold color that we know from jewelry and church relics and things like that. When they are nanoscale, depending on the specific size, but they tend to be red or other very, very bright colors, red through to purple. So they have very different properties when you make materials at the nanoscale. So some of the unique properties of nanomaterials, so they have a very, very small size. So this idea of being less than 100 nanometers means that they have access to all areas. And by all areas, I mean they can get into almost any organism, they can get into any part of our human body, they can cross through biological barriers in principle, they can get into the organelles of cells and so on. So they really operate at the same size lengths and size scales as viruses do. So, and we know how challenging viruses are in terms of their ability to adapt and respond. The large surface area I mentioned already is a hugely important and unique property. So it brings with it high energy and high reactivity, but also then a very high propensity to bind other things, to bind to biomolecules or other pollutants. They have properties of strength and lightness. So as I said, nanoparticles can be put into other materials, so into polymers or into plastics to make cheaper, lighter and stronger materials. And then their molecular properties, so this high surface reactivity means that they have a lot of potential for binding other things to their surface. So what that tells us then is that the key enabling length scales that make nanotechnology so exciting and offer potential to revolutionize how we care for our planet are also the potential risks from nanotechnology. So these same properties that we exploit for applications are also the ones that we have concerns about in terms of their risk assessment. So then, having a think through some of the risks from nanotechnology, I always like this picture just because it helps us to reflect that we need to keep things in proportion and not get overly concerned. But why do we do nanosafety research? What are the questions we're asking and what are the challenges we're facing? So first of all, the enormous variety of different nanomaterials is one of the big challenges. Another is that the same nanomaterial of the same composition may have multiple uses or applications. So if we take, for example, silver, it has antimicrobial properties. So it's very widely used in medical devices and things like that to prevent infection. But then it's also used in wastewater treatment. It can be used in household products. It's used in socks to make them less liable to smell over time and to reduce the amount of washing you have to do and so on. So a huge range of different applications from the same material. And this leads to challenges also because if you have some applications that are really important, like antimicrobial resistance in hospitals, but then the material is so widely used in everyday products, there's an increasing risk then, or an increasing chance for antimicrobial resistance to develop. So it's how we manage the multiple applications of the same material. And then, of course, responsible innovation, which is a hallmark of certainly all of the EU-funded, the European Union-funded programs in research, but also, I guess, a fundamental tenet for all research is that we should, in parallel to developing applications, assess the safety of them. So the ultimate goal of nanosafety research is that we would get to a point where we could group nanomaterials based on the properties they have and how those properties link to a biological impact. So there have been some estimates that there are more than 30,000 different nanomaterials being developed, excuse me, in labs and companies across the world. And to test each of those individually for their safety would take more time and more money than we could ever possibly afford. So what we want to do is get to a point where we can identify groups of materials based on their uptake behavior or their toxicity or their the sorts of applications they have. So materials could be grouped on their physical chemical properties or on their biological properties. The idea then would be that you would have to do less testing on new variants because you could predict their properties from previous versions. Another big challenge with nanomaterials, though, is that they're not static. They are very, very dynamic and they respond to their environment. So in the center of this picture here, we have sort of an idealized nanoparticle. And that's possibly what it looks like as we make it in the lab, assuming that we were going for a spherical particle. But once we put that particle into a formulation, into a product, or into the environment, or in contact with cells or anything else, it starts to change, it interacts. So it can interact, for example, by changing its agglomeration state. It can start to, if it's not that stable in the new environment, the particles can cluster together. So now you have different sizes of clusters of particles. It can undergo chemical reactions with species that are present in the environment, so with chlorides or sulfides, for example, which can change the surface properties, or it can oxidize. It can lose some of its functionality or it can gain functionality. So if it binds to proteins or other biomolecules present, that then gives it a new identity that changes it from the synthetic identity to a biological identity. We can also then have changes due to other processes, so physical processes like dissolution. So that can lead to changes in the composition. So, for example, if some part of it dissolves faster than another, it can lead to changes in porosity or the size of pores or the surface area, but also then changes in size due to dissolution, or even changes in shape. So we'll often have processes where the particle will start to dissolve, for example, and then potentially reprecipitate as something different. Just as an example of that, these were cerium dioxide nanoparticles. They started off as 3 nanometers, so this is very high resolution and you can see the crystal structure in them. But when they were incubated over time in a soil pore water, so water extracted from soil that had phosphorus in it, they changed very, very dramatically, both in size. So they went from 3 nanometers to around a micron, 1,000 nanometers, but also then changed and they had these spikes growing out to them, so they became urchin shaped. And obviously the toxicity and the uptake of these two different entities will be hugely different. And these spikes on these larger particles could, for example, puncture cell membranes or lead to things like frustrated phagocytosis, where if a cell is trying to take them up, attaching onto one of the narrower parts, and then isn't able to fully engulf the rest of the particle. So this is, for example, a process that happens quite frequently with carbon nanotubes or long rod-like and wire-like particles, where the cell is not able to take in the whole particle, but keeps releasing the inflammatory markers that tell it should be doing this. So this dynamic nature of nanomaterials means that they transform very much and very quickly, and that now the different forms will all have different effects. So I mentioned, for example, we can have chemical transformations of our particles, we can have physical transformations, and we can have interactions with biomolecules. And all of those can be happening in parallel. So it isn't always that only one of the processes will happen. We may well end up then after a few minutes with some particles that have been chemically transformed, some that have bound biomolecules, some that are a mix of chemically transformed and bound biomolecules, some that are still single particles, and some that are agglomerated. And each of these will then get taken up and interact in different ways, as shown sort of schematically here. So cells have a whole range, as you know, of different types of receptors that recognize different biological molecules, but also operate at different sizes. So phagocytotic processes will take up often quite larger particles, but clathrin-mediated endocytosis, for example, has an optimal size range around 30 nanometers. So depending on the size and the transformations that have occurred on the surface, these particles, these different forms of the nanomaterial will now bind different biomolecules and be recognized potentially by different receptors at the cell surface. The other thing we've got to think about then is that the new properties that arise as a function of the nanoscale. So I talked about quantum confinement. I talked about the different colors and forms and different crystal structures and things. All of these can potentially lead to new risks. And we need to be looking at those in the short term, the medium term, and the longer term. And the nanoscale particles have access to cellular compartments that larger particles or bulk particles, but also molecular entities don't have. So often molecules will behave quite simply based on chemical equilibrium. So the concentration inside an organism can never exceed the concentration outside, whereas that's not always the case with nanoparticles because they're going in by receptor-mediated processes. So they can actually preferentially accumulate in organisms. And if that happens then, so if we, again, taking the example of silver, silver ions are often considered to be more toxic than silver nanoparticles. But one of the problems that can arise is if an organism or a cell takes up the silver as a particle, and then once internalized, it degrades or dissolves, you will then end up with a much higher local concentration in the cells than you would have achieved by equilibrium. So it goes in as a particle and then dissolves. So you get much more in. And that's been called a sort of a Trojan horse effect, where the particle form carries in much higher concentrations than would have gone in through chemical equilibrium. There are a whole range of other challenges as well. So, so far I've sort of shown fairly simple spherical particles or relatively straightforward rods, but there are all sorts of other weird and wonderful and complex structures that are being developed. So, for example, dumbbell-shaped ones or flowers with different facets and different numbers of petals and sizes of petals, but also mixed components. So where you might have, a core or a shell, or part of it is one material, then another, then the first material again, and so on. So the growth of new structures is accelerating, and it's very, very difficult to even name those consistently and in an agreed way, never mind study their biological and toxicological impacts. Also, I've alluded to this already, but some nanomaterials will exist as they have a primary particle size, but actually in reality you will rarely get the individual particles, but you might get clusters of the particles, and these clusters might be different sizes as well. So this is a case of some industrial titanium dioxide, TiO2. So the primary particle size is 3 nanometers, so that's this tiny piece here. But actually, we can never, no matter how much energy we put into the system, we've never been able to get back to 3 nanometer particles. We always have them as aggregates ranging from 40 nanometers to greater than 300 nanometers. But then you might be presenting to cells a whole range of different things, where you've got some big clusters like this, that if it fell down onto a cell would actually cause physical damage. So basically just crushing the cell, compared to these smaller aggregates, which are small enough to be taken up through the receptor-mediated processes. There's also quite a bit of confusion in the literature as well as to what is an aggregate versus what's an agglomerate. So ISO, who are the International Standards Organization, came up with some agreed terminology, but it's not always used consistently. But technically, an aggregate is where the primary particles are covalently, so chemically and irreversibly bound and cannot be broken down. Whereas an agglomerate is where it's an environmental change that has caused the particles to agglomerate, and that usually can be reversed. So technically, most of the changes that I have talked about before, when they go out into the environment or they're released from a product, should be considered agglomeration rather than aggregation. Another challenge is, as if there wasn't enough so far, another one is that there are, for many materials, there are many different ways to produce the material, but yet they all are called the same thing. So this is just an example for silica, which is a very widely used material with a whole range of applications. And starting from the same starting material, there are a number of different synthesis routes to make silica. So through surfactant addition, and then you get ordered porous materials. You can do it through crystallization processes, which lead to different sorts of crystal structures, through dissolution and polymerization, gelling, dispersion, and so on, plus through pyrolysis, which is a heat-based process. So all of these have very, very different structures, but they are all silica particles. So what we often find is that the literature talking about silica particles or other particles don't necessarily provide all of the information on how the particles were made, which will provide some insights into what might be on their surface and what structure they might have. So all of that then is leading me to a set of properties of nanomaterials that might be prioritized in terms of understanding their biological impact. So the first property could be the dimensionality. So I talked about whether they're one-dimensional, which is with their a thin surface, whether they're two-dimensional, which is, for example, where they have spikes coming out of a surface, or potentially where they're three-dimensional, where again, membranes or nanopores or things like that. And again, whether they are long aspect ratio, such as nanowires, or small, low aspect ratio, such as particles, will matter a lot. So the idea is that if they're embedded or stuck on a surface, they will generally have less hazard and less risk than if they're free and therefore inhalable, or can pass into the body through the digestion system or through the nasal passageways or things like that. The aspect ratio appears here as well, that there's been quite a bit of work on high aspect ratio particles, so wires and tubes and things. And the reason for this is that one of the wonder materials of the past, asbestos, was nanowire type structures, or wire high aspect ratio materials, and they were found to lodge in the lung tissue but not be internalized fully, leading to asbestosis and silicosis and a rare form of cancer called mesothelioma. So one of the major concerns around nanosafety is whether carbon nanotubes and other engineered nanorods and nanowires would have similar toxicity concerns. The composition obviously will matter because I mentioned already that a whole range of elements from the periodic table can be made into nanoparticles. So for example, one of the major classes of nanomaterials are the quantum dots, and these are again very, very brightly colored materials, very small, usually less than 10 nanometers, and they're widely used in computer screens and in television screens. But they often are made from elements that are themselves quite toxic. So there's, for example, cadmium, selenium, or cadmium tellurium quantum dots. And the reality is that if the chemical itself forming the nanomaterial is toxic, there's a good chance that the nanomaterial will also be toxic. Whether the toxicity comes during its useful or functional lifetime in the product, or whether it comes at the end of the product's life cycle, so when the phone is thrown away and starts to break down and the cadmium is then re-released to the environment. So it's unlikely that something that was toxic to start with, or if the element itself is toxic, it's quite probable that the nano form of it will also be toxic. And then there's all sorts of issues with there's lots of strategies that are being developed for potentially reducing the toxicity of nanomaterials, which could be coating them so that there isn't any potential for release of the cadmium, for example, or mixed phase particles, and so on. And then another property that plays an important role is the uniformity and whether they're agglomerated or not. So again, if they're very well dispersed, they're essentially free and more likely to be taken up during exposure, whereas if they're agglomerated, or particularly the larger the agglomerate, the less it behaves like a nanomaterial anyway. So a whole range of features that need to be characterized and then potentially correlated with the biological effects. Added to that then, as I said, is this idea of the adsorbed corona of biomolecules. So when we take our pristine nanomaterial that we made in the lab and we put it into either a formulation or into cell culture medium to do an exposure, that pristine surface very quickly gets decorated with biomolecules from its surroundings. And if that's in a human tox scenario, it'll likely be some of the serum proteins, it may well be some membrane proteins and so on that bind, as well as other biomolecules, so lipids and small molecules will also be in there. But what happens is that our synthetic identity that we've now characterized gets altered and evolved by a biological identity. And it's these proteins and other biomolecules that are bound that determine which pathway the nanoparticles use to enter cells. This was quite an old study. This is about 10 years old now, but it's still nice to illustrate the point that in the absence of proteins, these were 50 or 100 nanometer polystyrene particles. And in the absence of proteins, an awful lot of particles were taken up by cells. These were A549 lung cells. And the reason that they were taken up is because the particles were so highly reactive that they basically just punched holes in the membrane, and then there was essentially, there was so much cell damage that they were easily able to go in. Whereas when there was this concept of the protein corona, when the particles were incubated in cell culture medium that contained serum proteins, so 10% fetal bovine serum, for example, which would be fairly standard in cell culture, then much, much less went in, but they went in through these very regulated cellular receptors. So just to wrap up this piece, that it's important that in order for products containing nanomaterials to reach the market, there needs to be there's a lot of work still needed. So we're still missing, for example, reference nanomaterials. We're still working in large part on updating the standardized tests for toxicity and ecotoxicity, which were developed for chemicals, and therefore assume solubility, and they assume oil-water partition coefficients, and they presume equilibrium behavior, which is not the case for nanomaterials. And I guess we're still working a little bit on what are the appropriate ways to describe nanomaterials to be able to link their physical chemical properties with their toxicity. And I mentioned also that it's important, because these materials are used in a wide range of properties, studying the form that we make in the lab or in the factory before it gets released is not necessarily giving us the right information. So we need to know, what's released during the use phase and what's released at the end of the life cycle in order to really understand what the risks are from the material. So just before I get into sort of some of the work we've been doing on how we test for toxicity, it's important, which I'm sure you all know having taken this course, is that everything is toxic at high enough dose. So to say that there's no toxicity from nanomaterials would be foolish, because we would need to talk about it in terms of a dose. So for hazard assessment, we're interested to find the highest dose where there are no observed adverse effects on the test species. So I'm going to spend a little while now, the next few minutes, talking about spions, which are a type of nanoparticle. They're a superparamagnetic iron oxide nanoparticle. So they're an iron oxide core, and they're usually coated with a polymer shell to make them more water soluble and more suitable for interaction with living cells. They're used in a wide range of things. They have useful magnetic properties that can be manipulated. So they're used, for example, in MRI imaging, for drug delivery, for hypothermia treatment, and so on. And although there was about 8 or 10 years ago, there was a number of products containing spions on the market. Many of them were subsequently withdrawn because there was a bit of a lack of efficacy in the clinic. And this related then to a lack of understanding around their cellular uptake and effects. So we were working for a couple of years. This is the work I'm going to present now over the next few minutes is from one of my former PhD students, who developed methods to measure nanoparticle uptake and correlate it with toxicity. So the methods need to be able to characterize the cellular uptake, the consequences and fate of exposure, and therefore help us to design more safer and more efficacious particles. And there's a range of approaches available. So the gold standard is always transmission electron microscopy, because that gives you extremely high resolution, allows you to see the cell ultrastructure and so on, but is hampered a little bit by the cost of doing it and the fact that you only ever see a very, very small part of the sample that you're measuring. Other approaches such as fluorescence and confocal microscopy give beautiful images and can have can provide quite a lot of information, but they are diffraction limited. So there's they can't actually see individual particles, for example. Other approaches such as atomic force microscopy can give you information on particles, and is increasingly being used to look inside of cells as well. Raman microscopy is also a useful approach. And then laser ablation, ICP-MS, so where they're actually ablating off layers of the surface and then measuring mapping the chemicals inside. So all of these are approaches that are available, but it's often difficult then to integrate the data that you get from the different approaches. So what we've been doing is looking at the different approaches. So with confocal microscopy, most people use it in fluorescence mode. So they add fluorescent stains and then use that to look at changes in the cell structure, or use fluorescence to label the particle, so then they can quantify how much particle has been taken up. But there are some challenges associated with the use of fluorescence with nanoparticles. So one concern is that if you fluorescently label a particle, are you changing it compared to the the unlabeled particle. There's also concerns that the label may come off, and just because you're quantifying the amount of the label that you're seeing, it's still not necessarily telling you that the particles are there, because they could have dissociated. So we were then looking at using the fact that nanoparticles themselves often reflect or bounce light off them. So we were looking to see, could we use confocal microscopy, but use the inherent reflectance coming from these, particularly metal or metal oxide nanoparticles, and use that as a way to correlate the uptake into cells. And even then go as far as looking at the co-localization, so where in the cell the particles are going by staining different organelles. So metallic and metal oxide nanoparticles in particular give a really nice signal. We've also then been comparing different modes. So confocal microscopy, as I said, it's diffraction limited. So we've also been looking at other approaches which give us a higher resolution, which I'll talk about in a minute. So this is an example of the sort of thing that we can do. These were cerium oxide nanoparticles, or the iron oxide, the spions, and these are the control cells. So we're looking at CTO, which is cell tracker orange, which outlines the cell, and we've got DAPI staining for the nucleus, and then we've got the reflectance channel for the nanoparticles. So we've overlaid the different channels. Then we can look at it we can look at the nanoparticles, and we can look at various processing steps to see, to establish the amount of particles that have been taken up. But what you see in some of those is you see what looks like clusters of particles. But one of the questions we had was, could we resolve those better to see, are the particles agglomerated, or is it just that the resolution makes it appear that they're all together? So one of the ways we've looked at is reflectance structured illumination microscopy, which again we can do in the reflectance mode, but it allows separation of clusters that were unresolvable by the reflectance confocal microscopy. So again, it just gives us a bit more resolution and a signal that in the fluorescent, in the reflectance confocal, appeared as one big blob. Under the SIM, the structured illumination microscopy, actually gets separated out into distinct regions of interest. So we can really get much better resolution in the images. Then we went on to see, could we actually correlate on the same sample and get the same cell and overlay the image we get from the TEM with the image we get from the reflectance confocal microscopy or from the SIM. So anyone who's worked with TEM will know that it is TEM is performed under vacuum, so you have to dehydrate the sample. So it's usually put in increasing amounts of ethanol to dehydrate it, and then it's fixed into an epoxy resin, and then with a diamond knife you slice your samples. So one of the challenges we faced in doing this was that as it's dehydrated, the cell shrinks a little bit. So it can be quite difficult then to find the same cell, because it might have changed shape and structure a little bit. So one of the ways we got around this was by using the gridded MatTek dishes. So you could at least find the same grid. So you would do your confocal microscopy first, note the grid of interest, and then do the TEM on the same grid reference point. We usually looked for cells that had some feature to them that would make them easily identifiable. So for example, there was two cells close together, or the cell had a particular flap on it, or something that would make it recognizable. And then we developed the code in MATLAB to overlay the images and restructure them. So this basically then allows us, so this is just sort of showing one of the things that we had to do was we had to re-stretch out the TEM image to account for the shrinkage that occurred during dehydration. And again, we used sort of an algorithm to allow us to do that. And these are just some examples of the overlay between reflectance imaging and the TEM imaging that allow us to identify nanoparticles, gives us then the ultrastructural information as to where they are. So, for example, that they're in lysosomes or early endosomes or so on. So the last little bit I'm going to talk about then is how having developed the method for tracking the nanoparticles, how we can then use that to look at uptake and toxicity and trafficking of nanoparticles. So again, confocal is really, really useful for nanoparticle visualization. And with the SIM modification, we get higher resolution, and we can then use that to so far what I've been presenting is in two-dimensional cells, so standard cell culture, but we've also shown that it works with spheroids, for example, so 3D cell culture. It gives us, so we've also been able to get quantitative information. So generally, uptake of particles by fluorescence would, we can actually count the number of particles because we can quantify or correlate the inherent reflectance from the particle with the amount of uptake. What we find is what we expected, in a sense, that the amount of uptake of the nanoparticles will depend on the incubation time. So longer times will allow more particles to enter. And this is very much related to the fact that it's receptor-mediated uptake. So each of the receptors will have a certain length of time that it requires to turn over. So once a particle makes contact with the receptor then undergoes the process of internalizing it and then is re-available for the next particle to bind. So we definitely find that the number of receptors on the cell surface is the limiting factor and their turnover time. So we were first able to do the counting manually, but that was obviously slow and could be a bit irreproducible and error prone. So we also then developed some algorithms to automate the process. So we take the images as we got them, we digitize them, then do some processing in terms of identifying the region of interest. We'd remove any cells that were only partially in the image, do some changes of thresholding and so on, and then get to the quantification of the nanoparticle uptake. So this works really nicely for quantification of the reflectance images, and we can, by adding as well the fluorescence, as I said, we can track in parallel both the nanoparticle quantification and the cellular quantification. So we can look at things like the impact of nanoparticle concentration on cell size, on cell shape, on the nanoparticle size. So we can also look at things like the potential for dissolution of particles once they've been internalized. We can look at nanoparticle intensity, which correlates with particle number and particle size, and the intensity then within certain areas of the cell. So we can correlate then the nanoparticle uptake with biological effects and plot it graphically then to allow different comparisons. So we've looked at, I mentioned the spions already, so they're used, one of their main applications was for heating of, they would be used for treatment of cancer, where they could preferentially locate to the cancer cells and then be heated up and give out heat that then kills off the cancer cells. So we were looking then at the mechanism of uptake and to see whether there was any specificity in terms of the cancer cell types. So we looked at a whole range of different cell types, including a HeLa just as our control, and then breast cancer cells and lung cancer cells and so on. And we looked at the uptake in a range of cell lines, and as expected found that it was both incubation time and incubation concentration dependent. So the more particles that were present, the faster they would get internalized. And in all cases, we had serum proteins present which was important. So we were definitely having the receptor-mediated processes. We also compared to macrophage cells. So these are the cells that are designed for uptake. They're specialized cells for taking up nanoparticles or for taking up foreign debris generally. And as expected, they turned out to be more efficient at the internalization. So what we found using this method was we can very quickly get an indication of the amount of internalization, but not which specific receptor-mediated process the particles were using. So again, looking at the different types of internalization, we were interested to understand whether the particles were preferentially using a clathrin-mediated endocytosis, a caveolin-mediated endocytosis, or a macropinocytosis. So we used a silencing RNA approach, so where we knock out the specific RNA that produces the proteins involved in the different pathways. So for example, for clathrin-mediated endocytosis, it's transferrin, which we knocked out. For caveolin, it was the CAV1 protein that we'd knock out. And for macropinocytosis, it was PAC1 and WAVE2 we used to inhibit those processes. So in each case, we compared pharmacological inhibitors and the siRNA, the silencing RNA, to target the specific pathways. We confirmed through Western blotting that we had indeed knocked out the relevant proteins, that they weren't being expressed anymore. And then we correlated that with the amount of particles that were taken up in each of the knockdown cells to understand the amount of uptake and which pathway was involved. So it's quite well known that siRNA is much more specific than the pharmacological inhibitors. So we were hoping that we would really be able to pinpoint which receptor-mediated process was used. And as I said, we confirmed that the silencing RNA used did inhibit the specific uptake. So we used the control chemical known to go in that route. So for clathrin-2, the control was transferrin protein. For caveolin, our control was the cholera-toxin B. And for macropinocytosis, the control substance was dextrin. And in all cases, we could see that when we had knocked out the relevant protein, the amount of uptake decreased. So when we knock out transferrin, we can see that the amount of uptake is reduced. When we knock out PAC1, we saw that the uptake of dextrin was reduced compared to the other proteins. So the knockdown worked well. And then we went on to look at how we could use this approach then to understand which pathways the spions were being internalized by in the different cancer cell lines. So again, we were using the orange, cell tracker orange to stay in the cell, the DAPI to stay in the nuclei, and the reflectance channel to look at the uptake of particles. So the white blobs that you could just think are speckles on the computer screen are not. They're actually the particles going in. So we can see that, when we had PAC1 knockdown, we still had quite a bit of particles going in the case of the breast cancer cells, whereas the caveolin, we saw very little particle going in. With CME, we saw some going in, and in the control, we saw particles going in. And similarly for the A549, we see in the CME knockout, there's still quite a bit of particle uptake. So then we compared we presented it in the same way. So we presented it as the percentage inhibition of uptake in each of the different siRNA cases, and compare that then to the percentage inhibition of expression. So in all cases, we're confirming that the expression of the particular protein was effectively knocked down. So what we found was that the HeLa and the A549 and MDA cells all included a CAV1 mediated uptake and a macropinocytosis. So the particles were able to go in through more than one mechanism. And this is really important because it means that if one mechanism is shut down or is poisoned, the particles were still going in, but by an alternative method. So they could use multiple uptake pathways. In macrophages, then, the mechanisms appeared to be dependent on AP2, which indicates then a clathrin route. And we also saw differences due to the particle size, the particle shape, and what their protein corona was, so what proteins had bound to the particles. We then went on to look a little bit at the final destination, so where the particles go following uptake. And again, we were able to use a range of different fluorescent markers. So the RAB signaling pathway, different variants of RAB localized to different organelles. So RAB5 goes to the early endosome, RAB7 goes to the late endosome, RAB11 goes to the recycling endosome, which basically then means that the particles will get transported back out of the cell. And we also use lysotracker for the lysosome. So we were looking to correlate where the particles were relative to to each of these fluorescent signals. It's important to know, though, that that visualization can be misleading. So this is the color merge. So this is the overlay of all the different channels. So the lysotracker channel, the DAPI channel, the RAB staining, and so on. Whereas if we deconvolute those and segment them, we can see much more accurately where the particles are. So with some of these, it might look like there's particles in the nucleus, but for example, when we deconvolute it, they're actually either in the layers above or below. They're not in the nucleus. So we also did various statistical tests, so Pearson's and Manders, to really understand the correlations between each of the different dyes from the endocytosis pathway with the particles. And again, we automated the process to remove user bias and to make it much more accurate. Both the cells and the fluorescent labels can be segmented and automated, and then we can look at, for example, the number of lysosomes that were counted and detected, and the number of lysosomes that were, so we can confirm that everything that we're seeing manually is counted by the software as well. And then we can really do the same sort of approaches where we can look at RAB5 for the early endosomes, RAB7 for the late endosomes, and RAB11 for the recycling endosome, and correlate where the particles are. So we could see a lot in the early endosomes by about 5 minutes, and less by about 30 minutes. And then by 30 minutes, they were increasingly present in the late endosomes and the recycling endosomes. And again, for the co-localization studies, the structured illumination microscopy offers us some advantages. Again, we can resolve some of these larger clusters and see whether they are one large lysosome, for example, or several just closely located lysosomes. Which again is important because one of the measures, for example, of toxicity could be lysosome size and lysosome acidity and things like that. So we can also then, using the reflectance signal of the particles, look to see what happens to the particles, so whether they degrade inside the lysosomes. So by looking at the co-localization over time, we can start to see whether there's increasing accumulation, or if we have a fixed pulse of particles, and then we track the cells. So we expose only for an hour or so, and then we track the cells over the next 24 hours. We can look then at the degradation of the particles, or, for example, the dilution effect due to cell division and so on. So really understand the fate of the particles. And again, if we do the correlative workflow, so incorporating the TEM also, and looking at the overlay between the three, we find that the combination of the three methods maximizes the information that we get and can really provide useful insights in terms of the effects. I just want to come back to one thing. So all of the studies that I've shown so far are performed under the standard cell culture conditions. So where we have cell culture medium with 10% seroproteins, where we present the particles, and then we assume that the system is static in a sense. But actually, there was a really interesting paper a couple of years ago from a group in Canada who showed that the cells themselves condition the media in response. So, for example, you have here the nanoparticle forming its protein corona and then engaging with some cellular receptors, and the early uptake, so those first particles that go in, cause the cell to respond by releasing other proteins or other molecules. So then those molecules that condition the media may have an affinity for the particles as well. So there might then be some evolution of the protein corona. So some of these secreted or conditioned molecules might come on, and some of the initial proteins might come off. So then they were suggesting that there might be a difference in the uptake of these intermediate particles, and then over longer times it might be even a different process. So there could well be that the initial particles are taken up by one receptor mediated process, whereas later on a different receptor mediated process is utilized because the particles have a different identity. And that's not something that's been taken into consideration really in standard testing or in how we go about understanding the longer term distribution of nanomedicines, for example, or the longer term toxicity of particles. So, oh, I'm stuck. Hang on. Why is it not moving forward? Ah, there we go. So that was really more or less where I want to end. Other than just to say that one of the major directions at the moment is nanoinformatics. So using in silico or predictive computational approaches to learn, to build on the data we have already for nanomaterials. To start then developing predictive models. So, but a really vital component of that is good data management, that the data sets generated in experimental studies need to be made available in a way that computational people can use them. So this is just one example of a very recent paper that used a data set from probably 5 or 6 years ago, because some of the more recent data sets are still not available. So this one was taking functionalized carbon nanotubes, so multi-wall carbon nanotubes, and developing an in silico model to predict the protein binding to the nanotubes based on their surface functionalization, and also then the cytotoxicity of the particles. Unfortunately, because it was an older study, the conditions for these two assays were not the same. So they had one set of conditions when they were studying the protein corona formation, and then they had a different set of studies different set of conditions when they were studying the cytotoxicity. So there isn't yet a way to correlate the proteins that are bound to the particles with the cytotoxicity observed. There are other data sets that do that, but this particular one, because it was an early data set, isn't possible to make that connection. So these are two separate models. One model predicts protein binding and the other model predicts cytotoxicity. And what this model did was it uses the idea of the K nearest neighbors. So it identifies the particles that have the most similar properties to the test particle of interest, and then it predicts its behavior based on the behavior of the nearest neighbors. So for example, if this was our test particle here, it identified 3 other particles as being its nearest neighbor in terms of the binding of this particular protein, and it predicted 7 other particles as being the nearest neighbors in terms of its toxicity behavior. And the idea then is that this reduces dramatically the amount of testing that has to be done. So I see my time is probably up. I'll just give a couple of quick conclusions. So there is no real evidence of very acute toxicity from nanomaterials, but there is quite a bit of more subtle effects. And certainly from a human perspective, it is the small changes over long times that are the things we need to worry about. There is still quite a bit of research and development needed to translate our understanding of two-dimensional cell behavior to more realistic 3D type structures that are present in our bodies. And as always, there's an increasing need for high throughput. As we understand the mechanisms and fate of internalization, we'll be able to get better then at predicting which materials will get taken up into cells and which will then start to cause toxicity problems. Regulatory testing is a really important area, but it's still very challenging. So at the moment, the standard tests, which as I said were designed for chemicals, are still undergoing revision. And that's particularly interesting in the EU context, because from January of 2020, ECHA, who are the European Chemicals Agency, will be requiring producers to provide data on their nanoforms, even though the tests are still being revised. So there's quite a bit of pressure then to revise the tests to take account of surface binding, agglomeration, non-equilibrium behavior, the dynamic evolution and the transformations that I talked about. There's still some debate on which form to test. Should it be the aged one, or the one that's released to the environment, or the one after it's been internalized, or how do we know what to test? And then of course the biological corona and their evolution and how cells respond to the presence of particles. So the first particles we're exposed to might be very different than the later ones. So how we take account of all of that, how we make sure our exposure conditions are appropriate, and how we follow and track sort of the exchange of the proteins as well. And then with that, as I said, I just wanted to thank Emily, who was the PhD student whose work I presented most of, and of course all of the funding that enables us to do the fun work, and to thank you for listening.
Thank you very much, Professor Lynch, for this very interesting presentation, lectures, and that summarize very well the theoretical part as well. But we see as well most of the up-to-date research is really, really interesting. Thank you very much for that. So we got a quick questions from Professor Evans. So thank you again for this very interesting talk. The question is, in many countries, we have government agencies that approve use of materials like drugs or pesticides. Do you think that we need the same for nanoparticles?
That's a really interesting question. And what I think is more likely is that each of those areas will have to have, so certainly for pesticides and for drugs, each of the existing agencies will need to have a set of considerations for nanoscale or nano-enabled versions of them. So I would doubt that a new agency would be able to cover the detail of all potential applications. But I think that certainly the challenge we're having in Europe is that, for example, the European Food Safety Authority have one definition of nanomaterials, the Cosmetics Agency has another definition, and the European Chemicals Agency has a different definition. So each of them have slightly different perspectives on what nanomaterials are. But I think there will in due course be convergence on that. And as the state of science solidifies, then I think it will be relatively easy to harmonize across the agencies. But I think, one of the things that's really interesting when looking in the context of pesticides, for example, is that the sorts of concentrations of particles that we need to be at to see toxicity are orders of magnitude higher than the concentrations of pesticides, because of course pesticides are designed to kill. So you very quickly get into the terrain of, oh, well, a nanoparticle isn't really a problem, it's the active ingredient. So Whereas if you're talking about something like a nanosilver coated baby's bottle, where the baby is going to be eating from that, that's their sole source of food for the first 6 months of their lives, that's a very different exposure scenario. So, and I would think would have a very different set of concerns around it.
Yeah, wonderful. Yeah, thank you very much. I might just have another quick question. Yeah. I just got a quick last questions. I believe we're running a little bit out of time. I'm more interested in the method development techniques.
Mm-hmm.
And would you think that because we got a lot of novel and advances in analytical tools, and would you see
Mm-hmm.
that the coupling between MODIs or matrix assisted laser ablations and ICP-MS would be able, an organic mass spectrometry, would that be able to provide better visualizations of either the organic contents, so the cells compositions, but as well the composition and the change of the structures of nanoparticles?
That would be the dream. Absolutely. Absolutely. And there's already some work towards that, with a range of different techniques. So correlating Raman spectroscopy, for example, which can look at changes in biomolecule structures. But yeah, I think hyphenation of methods really is the way forward, because otherwise you're not sure that you're looking at the same region of the sample, the same consideration. So I think the pace at which the analytical methods are developing is very rapid now, because some of these key questions have been identified and there's now a market for these advanced methodologies. So yeah, I think it's a really exciting space.
Yeah, thank you very much. Yeah, because here at Trent University, we got our water quality center, and I think we are a bit well equipped for this kind of hyphenation of the techniques. Yeah. Yeah. Thank you very much. So I see that I got Sarah who raised up the hand. I don't know if I think she got a question to you.
Yeah, just a quick one. It's in regards to what you were discussing about the nanoparticles interacting with the environment. So obviously different types of particles with different compositions will react differently, but I was wondering, say you had just some set of like nano silver, would individual particles ever interact differently, like react differently with the environment and come up with these secondary products that are different from each other?
So if there were different sources of silver, for example?
I was just actually wondering, like, if it was the same source of silver, like if one particle next to another and one has reacted differently.
Yes, so that's a hugely interesting question as well. And at the minute, there aren't all that many techniques that can do particle by particle analysis. So we assume in large part that all of the particles within a particular batch or a particular type will be the same. But in the same way that we think about a size distribution, so we know they're not all identical size, there's a plus or minus in the size. I'm absolutely convinced that there would be differences within a batch of how much charge is on the surface or how efficiently they've been coated by the capping agent or things like that. So yes, I would think there's a very real possibility that there could be within a single batch differences then in terms of what biomolecules they bind to, how they might speciate, how much they might bind or interact with sulfates or chlorides or other things. So absolutely.
All right, thank you very much.
Awesome, thank you very much. I believe that we are a little bit out of time, so
I'm sorry about that. I obviously ran over.
It is not a problem.
Full screen, I couldn't see the clock on my computer.
It's not a problem at all. Thank you very much, Professor Lynch. Actually, this lecture from Professor Lynch closed a series on risk assessment that we are having now. So thank you very much again to you, Professor Lynch, for your lectures, and thank you very much for all of you for your interest in our series. And I hope to have more of your interest in our future events. So we're having something else on air pollutions that's gonna start next month, and then we have something else as well on carbon innovations that are gonna start likely before the summer. So thank you very much again, and wish to you all the best for your future endeavors. Thank you.
Thank you. Bye-bye.
Bye bye.